Welcome to another episode of Science Talks, a conversation hosted by the University of Arizona's BIO5 Institute. My name is Amy Randall Barber. Today, I have Caroline Mosley hosting with me. Caroline is the Senior Manager for Communications and Content here at the BIO5 Institute. We interviewed Caroline in an earlier podcast, and we found out that she was once a scientist studying invasive species in the Great Lakes, but she is now passionate about science communication and is telling the story instead of doing the science, which is very interesting. So Caroline, thank you so much for joining me today.
Thanks, Amy. It's great to be here. I'm very excited to talk with our guest today and learn more about her amazing work in cardiovascular disease. Understanding the role of cardiac myosin-binding protein C in muscle function can offer insight into cardiovascular disease. Mutations and abnormalities in this protein can lead to defects in the cardiac system, including heart failure in adults. So today we are joined by Dr. Angie Greenman. She's a 2024 BIO5 Postdoctoral Fellow in Dr. Samantha Harris's lab in the Department of Physiology at the University of Arizona College of Medicine, Tucson. Angie studies molecular mechanisms of cardiomyosin-binding protein C that will hopefully lead to better outcomes for understanding muscle function and cardiac disease. Angie, thank you for joining us today.
Thanks for having me.
So we always like to get started with just kind of a fun, lighthearted, simple, rapid-fire style type questions. So I will ask the first one. What do you prefer, coffee or tea?
Definitely coffee.
Okay, and then of course tell us what your favorite coffee drink is.
So I lived in New Zealand for a while.
Ooh.
And I got really into flat whites there.
That's cool.
So what is the most adventurous thing you've ever done?
Such a funny question because I feel like I'm not very adventurous. I'm kind of afraid of heights, but one time my friends convinced me to jump off a cliff into this little beautiful waterfall area, and I thought, I had such a big adrenaline surge after that. I still remember how much fun that was. But
did you just do it once, or did you go back?
I just did it once. I couldn't do it again.
I feel like if that's not only adventurous, but also overcoming a fear.
Yeah.
Which makes it even more adventurous, I would say. So what is the best piece of advice you've ever received?
It's a good question. Regarding now in my life as a postdoc, probably that don't rely on just one mentor, but reach out to other people and build your network of mentors. So not one person can give you all you need in terms of mentorship as a postdoc.
That's good advice.
Yeah, I think that's just life advice in general.
Yeah,
Have
exactly.
a good community. That's great.
Awesome. Well, thank you. So we're always curious as to our guests' backgrounds, who they are, how they got into science in the first place. Can you tell us how you became interested in science?
Yeah, my mom was a pharmacist, and so I feel like always exposed to health sciences. And for a while, I wanted to go into the clinic, so I did the biology pre-med route, and then I was like, I should join a lab. Like, that'll look good on a med school application, right? That's what people do. And I joined this lab that looked at skeletal muscle function, and I just kind of fell in love with it. I think at the time I thought people who get their PhDs are like insanely smart weirdos, savants, and I'm sure there's some of us like that, but it made me see science being a scientist was a feasible career goal for me.
Well, that's awesome. Yeah. You were in New Zealand for your PhD. Can you tell us about that? That sounds exciting.
Yeah, it was really fun. I kind of just got offered to go there. So I was at the time I was doing my master's at the University of Wisconsin, and I was finishing up writing my thesis, and my mentor was gone in New Zealand, starting a new collaboration. And he didn't respond to any of my emails about questions and concerns about my thesis, but he responded saying, hey, do you want to move to New Zealand? Like, they need someone like you to do a PhD in this lab. And I was like, oh, yeah. That sounds amazing, moving abroad, and it was a really great opportunity. So you can tell by my accent, I'm American, and so it was the first time living abroad, and even though Kiwis speak English, it's a different dialect and just different cultural norms. So I think a real strength of me doing my PhD there was a better emphasis on the balance in terms of the work-life balance. So I really appreciated that when I reflect back on that.
Oh, wow. Okay. Is it just different the way that they are there with that?
Yeah, it's really, everyone goes home and they and they like stop working, you know?
Oh, wow.
And they won't answer your emails after 6 or something. And
They just have good boundaries.
yeah, if there's a holiday, they're not at work, right? Like that's your day off. So of course a PhD still, you have some longer days, but in general, I saw a better balance in terms of PhD student work-life balance. Yeah.
That's awesome.
That's so cool. Do you still have collaborations in New Zealand from your PhD?
Yeah, a bunch of my mentors are still there. We're not actively collaborating, but would always be open to the idea, of course. And I hope to visit in the next 2 years or so. It's been too long. Of course, I have friends there. And
Yeah. Nice. So what was it that brought you to the U of A and to BIO5?
Yeah, so I knew I wanted to do a postdoc. I'm really interested in getting a tenure track position, so some sort of combination of teaching and research down the line. And I guess for those who are listening, don't know, you kind of need extra training in between a PhD and becoming an assistant professor. So yeah, it came down to like the science, what kind of science I wanted to do, and I knew I really want to stick with measuring muscle function, and so that kind of narrowed it down for me. And I also knew I wanted to have a mentor who was a woman in science, and so I've had great mentors who were men, but I kind of wanted to diversify my mentorship team. And yeah, it came down to Dr. Harris's lab was one of that kind of fit all these needs, and I really liked Tucson as well, and so did my partner. So yeah, we ended up reaching out to her, and it all worked out, I guess. It seems really simple when I say it now, but of course when you're finishing your dissertation and you're like, how do you have time to do these things? So yeah.
Isn't it amazing too? I find that the researchers are very responsive. You wouldn't think you would think they're so busy and your email's going to get lost in their sea of emails, but no, they are quite responsive.
When someone reaches out to you and says, I like your work, we should work together and do more cool work. Normally someone's going to reply.
Yeah.
So true. So can you tell us a little bit more about your current lab and the Harris lab and their goals?
Yeah, so Dr. Harris is really focused on this one protein, which is in muscle. So it's in skeletal muscle, heart muscle, but we primarily focus on the heart. And so just for a little background, heart muscle is the smallest functional unit of muscle is called a sarcomere, and there's these different filaments that kind of overlap with each other. And these filaments are made up of proteins, and one of those proteins is the protein we study in our lab called cardiac myosin binding protein C. So these filaments are important because they slide past one another. Actin and myosin are mainly the two proteins doing this, and they produce force. So, flexing my bicep right now, I feel like I'm stiffer. Those are literally the myosin and actin interacting, kind of interlocking together. So what we find is when we get rid of this protein, myosin binding protein C, there's a lot of dysfunction, there's disease, and it all goes down to that individual sarcomere level of dysfunction.
So how do her lab goals kind of align with your career goals?
Yeah, so I guess getting back to the previous question too of why her lab is, I got to, I was already using similar tools to measure muscle function, but she had a few more protocols that I hadn't used before, so I became more sophisticated at these measures. And I'm also learning a lot more molecular biology techniques, so I purify a lot of the protein, C protein for short. And so that takes cloning skills and working with E. coli, things like that that I hadn't done before in my PhD work. So it's actually really fun. I feel like I'm kind of like a mad scientist in the lab, like creating new things, except the goal is not to, destroy the world, but to help the world.
A much better goal.
Yeah.
So could you tell us a little bit more about your current project that you're working on?
Yeah, so myosin binding protein C is this long protein that can interact with both actin and myosin, but when that happens, we don't understand. And it also seems to affect how fast contraction can happen, either in a positive or negative way, which is really confusing. You would think it could only do one, but it seems to be coming down to when it interacts with actin or myosin, it has a totally different effect on contraction. So I'm studying just the middle portion of this protein, and previously people thought it just kind of linked the two ends together, and that was all it did, but it seems to interact with actin and myosin directly or indirectly affecting muscle function. So in hypertrophic cardiomyopathy, that can lead to heart failure. Some patients have mutations within this region of the protein, and we don't really fully understand how that mutation links to their symptoms. So I'm trying to better understand. Currently, I'm really focused on just normal physiology of the protein, and then the goal is to focus on those specific mutants that we see in the clinic. Mm-hmm.
So was this middle section not really being studied before? Or how did you kind of discover that there is something maybe more going on there?
Yeah, it had been studied a little bit, but it's really hard to study with the models we have. And so what was also really cool for me joining Dr. Harris's lab is she has this mouse model where we can literally cut and paste our protein. And so we can paste back different segments of the protein, so certain deletions or substitutions or mutations, and that can tell us which parts of the protein are important for different aspects of muscle function. So before this cut and paste model, it would be really hard because you'd have to kind of like in vitro or just look at tiny segments of proteins, and so that doesn't really always translate to what's happening in your heart right now, right? So this model provides us the ability to test these questions really specifically. So people were interested in the middle domains, but I would say it was just really hard to design a strong study for those questions.
Oh, that's interesting. So you kind of covered this, but I'll just ask it if you want to add. So can you break down and explain to us why cardiac myosin binding protein C, what it is and why it's so important?
Yeah. Well, it's one of my favorite proteins now. So in patients with hypertrophic cardiomyopathy, a lot of the mutations lead to the truncation of the protein, which essentially you can imagine it means that the protein isn't there or only a segment of the protein is there. And when that happens, they have enlarged hearts, they have symptoms like shortness of breath, they have increased risk of sudden cardiac death. So it's just one protein and you can survive without it, at least in your heart, but it can lead to a really poor quality of life.
And again, you kind of answered this, but what is hypertrophic cardiomyopathy and how does that protein play a part in it?
Yeah, so hypertrophic cardiomyopathy is a genetic disease, and it's only known to be within the sarcomere. So any of those sarcomere proteins, some of them, myosin and myosin binding protein C, are the two most commonly known proteins that get mutated, and then we find it in the clinic. So what happens when these proteins are mutated? It seems to lead to this hypercontractile state of your heart. So I guess the analogy is that you're just sitting like we are here, but your heart is beating as if it's going for a jog. And so over time, if you have that consistent stressor on your heart, it's going to remodel, it's going to enlarge, which isn't always a bad thing, but with other things like fibrosis, which is essentially the stiffening of the heart, a lot of times these patients can have a hard time pumping enough blood out of their heart to the rest of their body, getting oxygen to your cells. And they can also have a hard time relaxing their heart, which is a really important aspect that gets overlooked a lot too.
Yeah. So how common is hypertrophic cardiomyopathy? Is it? I know it's a cause of heart failure in adults, but is it? How common is it?
Yeah. So estimates are around 1 in 200, as high as 1 in 200 people actually have it. So I didn't make this point, but a lot of folks are undiagnosed because a lot of times they don't have any symptoms, or their symptoms are really mild. It might seem like asthma, or I just haven't been going to the gym anymore, so that's why climbing up a flight of stairs is hard. And it can even be slowly progressive. So you might just think it's aging, and you might not realize, it's not normal to feel lightheaded after this amount of activity. So
wow. That's really interesting. I didn't realize it was so common, but I always feel like it's kind of amazing how the body can adapt. And I guess body and mind. So, wow, 1 in 200. So going back to the, you're a 2024 BIO5 Postdoctoral Fellow. So as part of your fellowship, you hope to travel to the University of Copenhagen and advance your laboratory skills. Can you just tell us more about that and what you kind of hope to experience?
Yeah, I'm super excited. So we have a collaborator, Dr. Julian Ochalo at the University of Copenhagen, and he has developed a model where he can study the energy rates of the myosin heads. And so I've kind of hinted at this, but in hypertrophic cardiomyopathy, those heads are usually in this hypercontractile state, and they're probably using up a lot more energy than they really need to. So it's really energy inefficient. And so we suspect that if we cut out myosin binding protein C in our different models, and we would expect to see that it'll be using up energy faster than ideal.
So you mentioned that, so you'll be learning the techniques that your collaborator is using in the lab. That's the main.
Yeah, that's the main goal. Also, get to give a talk to his Department of Physiology, and I'm really excited because I'm really focused right now in the Harris lab on one protein, but my background is more of looking at the effects of disease on the whole sarcomere. So he is more interested in a disease-based approach in his lab. So it'll be nice to collaborate with, just thinking through new ideas as well.
Very cool. It was also mentioned that you use fluorescent microscopy techniques to show a relationship with muscle function. Can you talk a little bit about that?
Yeah, so that's what I was trying to get across. Ochalo's lab uses fluorescent microscopy in order to estimate these different energy states of myosin. And so essentially what we do is we isolate these little bundles of muscle, and then we expose them to this fluorescent ATP being the energy source. And what we can do is then wash it with just regular ATP, which outcompetes for that fluorescently labeled ATP. And then you can just watch the decay of the fluorescence. And so there's 2 general states. I mean, this is a little bit controversial, but there's generally a state in which it's using up energy slowly, and then a state where it's usually using up its energy really fast. So we call that like the super relaxed versus the disordered relaxed states. So I'm going to quantify those 2 in different cut and paste approaches.
Are you hoping is it possible to bring those techniques back to the Harris lab, or do you need special equipment to do that?
That's the goal. So the goal is I'm going to collect some data while I'm there, but also learn the approach well enough that I can set it up in the Harris lab and hopefully my own lab someday. So, yeah.
Very cool. Thank you.
Yeah.
Can you give us an example of something in your project or in your lab that made you super excited, like a new discovery or a new anything that just got you super excited?
Yeah, I mean, I'm really excited right now, my data set. I just presented some work at a conference and got some really excited responses. So the middle portion of the protein, it seems to be really important for kind of moving one end of the protein away from actin. And so without those domains or that segment of the protein present, it just keeps activating actin, which means the probability of myosin and actin binding together and sliding, producing force is higher, which sounds like a great thing, right? You want more force, but in the heart, you also want to relax. So it's kind of this trade-off. So really excited. I'm preparing this for a publication soon, and
Oh, cool.
the cells just behave so weird. It's normally in science you have like a slight difference, but it's a stark difference when you're working with these.
Interesting.
Wow. You kind of alluded to it earlier, but you are interested in becoming a teacher or a professor as well as a researcher.
Yeah.
I want to comment. I think you're going to be a fantastic professor. You have been able to describe all of this stuff very, very well for our listeners, so thank you.
Thank you.
Is there any particular mentor that you've had that inspired the idea for you to also become a professor?
Yeah, I would say my first mentor, Gary Diffie, the PI from Wisconsin that I did my master's with, he was really influential to me. He, this was the lab I joined and thought, I'm just getting credits for med school. And he kind of showed me that science can be really fun, and but you can also have fun outside of science. And he was just a really great role model. And I guess I hope to continue to get better at presenting, but he taught me a lot of my presentation skills of, don't go too fast, like walk them through it as if they're your friend, right? And so, he's a great mentor, and I hope to keep learning from him.
So what is your why?
Yeah. I've noticed my why is always changing a little bit. I think when I started, especially in my master's, I was studying the effects of cancer on muscle function and muscle wasting. So this is called cancer cachexia. And at the time, my dad had cancer. And for me, that was so important. My goal as a researcher was to better understand how these diseases affect muscle function in hopes that, we can understand how we could target that for therapy purposes and whatnot. And not just for cancer patients, but now I'm, focused on heart disease, and I've looked at the effects of diabetes on the heart. So just realizing that you can really help individuals with the work you do is really powerful. I do notice as I get more senior, I also feel like a why is I can make a really big impact as a mentor myself. And I've seen good mentoring, I've seen bad mentoring, and I feel like I can be someone in the room who can help, be a better mentor, which to me is just being approachable and friendly and kind and, a good listener. And and I've hinted at this, New Zealand was a great influence on me, and focusing, students, obviously, we know this, PhD students have a high rate of mental health issues, and I think sometimes the pressure they face from their mentors just really doesn't help that. So that could be a whole conversation in itself, but I see myself having two big whys now.
Those are great whys, and I think if we researchers like you studying, heart muscle and different kinds of diseases and really focus on mentoring, I think we're going to be okay in the world. So.
I hope so.
Yeah, absolutely. Well, Angie, thank you so much for joining us today and being our guest. We really appreciate having you and getting to know more about you and what you do.
Yeah, thanks for having me. It's been really fun.
Thanks to our listeners for tuning in to another episode of Science Talks. Continue the conversation with us next time as we learn more about the amazing science happening at the University of Arizona's BIO5 Institute.